what we can see is a small reduction, about 15%. It's quite reproducible. Small reduction in microglia in the brain. Okay. But this makes a very strong prediction, and that is we should be able to cure this disease by a blood transplant. Okay? What you do is radiate the mouse, put in blood or bone marrow cells from, say, another mouse. It repopulates, and now if that mouse doesn't have a HOXB8 mutation, it should cure the disease, if that's the cause. Okay? So what we're saying is we can cure a neuropsychiatric disorder by a bone marrow transplant. And the answer is shown here. First of all, if we do a bone marrow transplant and make the green cells, do we see still cells in the brain? And we do, the green fluorescent cells in the brain that are now coming from, because we've labeled those bone marrow cells green. Okay? So that says that we do the bone marrow transplant, it can get to the brain. And then the actual experiment is shown here. So here we're looking at about 3 weeks after transplantation. It started removing its hair. If I showed you this a couple of weeks later, it would start having lots of lacerations. And now, by about 4 months, which is about the time it takes to graft the bone marrow, now all the lacerations have been healed, all the hair has grown back. And further, if we look at the time spent grooming, here's the wild type, the normal, here's the mutant, and now here's the rescued, and now it's back down to normal. So both with respect to how much time they're spending grooming, as well as all the lacerations and removal of body hair, they're completely cured by a bone marrow transplant. Okay? We can do the opposite experiment. We can stick mutant bone marrow in a normal mouse, and now it has full-blown pathological grooming difficulties. Now, there was a complication, and the complication is because I told you HOX genes are very complicated to do many things. And so often when we knock out a HOX gene, we see many things happening. And another thing we see in these mice is defects in sensing pain. These are nociceptive defects. Okay? And for example, what we can do is put a mouse on a hot plate, and then we can measure how quickly does that mouse jump. Okay, and normally a mouse jumps very quickly as soon as it it's uncomfortable. Okay, so that's called essentially a hot plate test. So here we're looking at the spinal cord. So cutting right across here, and this is the spinal cord right here, and this is what we call the dorsal aspect of the spinal cord. And what we've done is to have a marker just for interneurons. Okay, and what you can see is, if you look carefully, this pattern would look to you differently from this pattern. It's not quite as big, it's more disorganized. Okay, but it's better if we actually look at particular markers to look at specific interneurons in the spinal cord. And that's shown here. So here we're looking at CGRP, which is a marker for the incoming neurons that are actually sensing the pain. Okay? And then this is calbiden, calretin, are actually labeling the interneurons themselves at the dorsal aspect of the spinal cord, and they make nice layers, lamina 1, lamina 2. And what you can see in the mutant, so here's normal, what we call wild type, and this is mutant. You can see that there's a lot less, and they're also more disorganized. They're not nice layers, essentially. They're more random, essentially, with respect to where those cells are. Okay, so that's what the HOXB8 gene is doing in the spinal cord, and why it has insensitivity to pain. And we can measure this. Here's the hot plate test itself. And what you can see, mutants take about 3 times longer to react to the pain than do normal mice. Okay? So that's a defect. Now, the first indication that we could separate what we call the nociceptive, the pain reception, from the grooming, pathological grooming, was in the rescued animals themselves. So here's the wild type, here's the mutant, and here's the rescued. But now we're looking at hot plate. Okay, so I told you that bone marrow transplant completely rescues the pathology with respect to grooming behavior, but it doesn't fix essentially the hot plate. Okay, it's still insensitive to pain. So that told us we could possibly separate those two. And one way we can do that is now to inactivate the gene in the very specific cells. We can inactivate it in the spinal cord, and that's what happens. We can inactivate it in the bone marrow, and that's what happens. Okay? So that's shown here. So Tay2 allows us to look at this in the bone marrow. We inactivate it. First of all, we show that now cells, if we use Tay2, they will label those cells, so they're getting to the brain and so on. And now here's the actual experiment. So we've inactivated HOXB8 only in bone marrow, but what we can see is they start removing the body hair. If we look to see how much time do they spend grooming, they spend about twice as much time as controlling. But if we look at the hot plate, they haven't changed, okay, compared to the siblings that don't have that defect. So it's affecting essentially grooming, but not insensitivity to pain. We can look at the actual spinal cord itself, and we can see here's the wild type pattern, here's the mutant pattern, and here's Tay2, and that's like the wild type pattern. Okay, so we're not affecting the spinal cord itself. We can do the opposite experiment. We can activate it in the spinal cord, and that's what happens. And for that, we use C8, which is a sister gene to HOXB8. It looks identical in the spinal cord, shown here. Here is simply showing you where that gene is expressed. Here what we can see is how many green cells do we get in the brain if we use HOXC8 instead of HOXB8. And it's 15-fold lower. There's a few cells that are green, but not very many. It also labels endothelial cells, blood cells, and therefore that's why the blood vessel cells, and that's why you're seeing cells green there. So those aren't microglia, but these are microglia, but they're 15-fold lower than if you had used HOXB8. So that allows us to use that to inactivate it more specifically in the spinal cord. And here are the results. Here is a perfectly normal mouse. It doesn't affect grooming behavior. Here's looking at time spent grooming. It's not different from control, but now it is completely insensitive to pain compared to the mutants. So that has allowed us essentially to separate the two and know that HOXB8 in the bone marrow is responsible for the grooming defects. So here we'll finally come to the end. And the reason this is of interest is, first of all, I've shown you that it's true for this particular behavior, trichotillomania. Trichotillomania is actually quite common. 2 to 3% of all populations tested all over the world have that particular defect. Okay, and what we're saying is that microglia is responsible for it, but it may be much more general than just that disease itself. If you look at any neuropsychiatric disorder, monopolar disorders, bipolar, autism, schizophrenia, I've already mentioned OCD, obsessive compulsive disorder. If you look at even Alzheimer's, all of them have been known to have defects in the immune system associated with that disease. But it was never clear what the association meant. Okay? Are you depressed and therefore your immune system isn't working well? Or is your immune system not working well and therefore you're no more likely to get now depression? Okay? Cause and effect wasn't clear. And what we're saying is there's a direct correlation between the immune system, microglia, and behavioral output. That a defect in the immune system in the brain can result in defective output. Okay? So it could be extremely general. How could microglia be controlling it? We don't know. Many models, for example, one thing that microglia produce is cytokines. This is the actual circuit that's involved in generating OCD and OC spectrum disorder. It's essentially what the basic ganglia, prefrontal cortex, thalamus. It's that circuit that's involved in all repetitive motions, essentially. OCD is essentially a defect in repetitive activity. It's repetitive activity gone awry. You continually do that particular thing rather than doing it and then forgetting about it and going on to the next thing. So that's that circuit. It could be cytokines are producing and activating that neural circuit. But there's something more interesting. Here we look at the microglia, and they sort of look like neurons. They have these long projections sticking out. And it turns out that these projections are being waved all the time in the brain. And in fact, you can calculate, because there are a lot of microglia in the brain, that in 1 hour they can scan the entire brain surface. just with these projections, moving around. But if you actually look at them, they're moving around, moving around, moving around, then all of a sudden they stop. And then they stay and they're fixed there. And then you can ask, well, what are they doing when they're stopped? And what you find is that they're actually surrounding a synapse, the connection between neurons, which are involved in propagating a signal. So they're actually engulfed that. You can actually see the membrane going all around the synapse. So that may tell us that this may actually be communicating with that neuron and that circuitry, and thereby directly controlling the activity of that circuit. So now we can start testing those kind of hypotheses. Is there a direct talk between neurons and microglia, and that gives rise essentially to these pathological